So the idea of this second part is to move from the function of the LHB in encoding aversive states to move it to its potential role in at least aspects of addiction or related to addiction. So what I wrote here is kind of to give you the context of it, where the idea is to have withdrawal-driven depressive states and have an overlook from the lateral abenula rather than from, let's call it, more regular circuits that have been implicated in addiction. Why is it? So this is mainly some of the work that we are carrying in the lab, but what has been done that set a little bit the ground to implicate LHB function in addictive behavior. So some papers have shown the following. First of all, cocaine, general psychostimulant, acutely, so while the mouse is sleeping and anesthetized, when you administer cocaine IP, that can bidirectionally modulate lateral abenular neurons activity. So first hint is that an acute injection of cocaine changes the activity of these neurons. Then, it has been shown, that the CFOS activation in the lateral abenula can occur after cocaine treatment, both an acute injection and even a chronic injection. Second hint. Then, back in time in 2002, there have been some papers suggesting that during a chronic treatment of cocaine, there was some of a loss of axons from the lateral abenula to the midbrain. But this was done with extremely high dose of both cocaine and amphetamines that probably triggered this degeneration. And then a couple of papers suggested that stimulation, indeed using the brain stimulation type of approach, could reduce drug seeking, but nothing was done in terms of what cellular modification can the cocaine intake induce in the lateral abenula, whether it would be this done in an acute phase or rather in the withdrawal phase. And more, what type of behavioral readout will be driven by these changes in the lateral abenula. So in the lab, we try to tackle this question. So here is a picture built by Gary Aston-Jones and Steve Mahler, where they looked at in which conditions, say here, Q-induced restatement or extinction or no behavior or specific activation, in which phases there was any FOS activation in which region. And so all these regions, like the VTA or the tail VTA, or the BNST here, but also the LHB, all pop out, suggesting that they could be activated, but not necessarily in a parallel way, and they would not encode the exact same thing. So this led us to kind of predict that some region would encode for the initial rewarding aspects, and other region would encode for rather withdrawal-induced negative states. So this is work from Tom Joo, and basically what you see, what they did here is they were passively injecting animals with cocaine. And then they looked at CFOS expression in the lateral abenula, and then what they did previously was to inject this cholera toxin B in this rostromedial tegmental nucleus, which is the GABAergic nucleus receiving the terminals from the lateral abenula. So in this way, they could tract back these lateral abenular neurons. So what they see is that they injected the retrograde in the RMTG and in the VTA. And the finding was the following. They find that the CFOS expression in the lateral abenula was mainly on those neurons projecting to the RMTG, but not VTA. So the first hint we have here is cocaine can activate LHB neuron or downregulate its activity. You see a bidirectional modulation. But you see this CFOS expression only in a specific subpopulation of those neurons going to GABA neurons of the midbrain. So, in other words, this desynaptic pathway seems to be implicated in some of the cocaine effect. Now, what's in there regarding the functional evidence? So this is, again, Tom Jewell, where he took these animals, anesthetized the rats, and he recorded single unit activity from LHB neurons. And what he sees is that there is a bidirectional modulation of these neurons over here, and then what he sees is that there is a rebound or a biphasic component that is an initial inhibition and a long-lasting excitation. So this is over about 30 to 40 minutes. So what is it due to this inhibition and what is it due to this excitation? This was still unknown at the moment. But it was already pretty curious to see, well, if we look at this late response, and we think in the order that LHB increases the activity of the LHB, that cocaine increases the activity of LHB neurons, that would impact negatively on dopamine neuron activity, right, via the synaptic pathway. So there is already a suggestion that hyperactivity in the lateral habenula would encode for something that is negative rather than positive by inhibiting dopamine neurons. So if that was the case, we wanted in the lab to understand whether there is any correlate at the synaptic level for LHB function according to where this lateral habenular neuron project. We wanted to understand whether the cocaine experience would lead to any sort of changes, driving either an hyperexcitation or an hyperactivity here in relationship to the downstream pathway where lateral habenular neuron was sending their axons. So we started trying to developing the idea that cocaine-driven negative states require adaptation in the lateral habenula by orchestrating midbrain structure. And we got interested in a basal ganglia projection, and we wanted to know whether these lateral habenular neurons receiving the basal ganglia projection will be implicated in controlling midbrain neuronal populations. So we started off simply with a simple paradigm that we knew it would drive behavioral changes, an increase in locomotor activity classically seen as a sensitization, and then afterwards leaving these animals with no access and no drug at all. So basically we submitted animals to a series of cocaine injections, IP, and we checked, okay, the regular parameter like induced locomotion, for example. What we find in this animal is the following. These are experiments done in vivo, anesthetized mice. And the recording site is here in the LHB, but what we were doing also was to try to identify neurons in the lateral habenula that would project to the RMTG. So we wanted to understand whether any change happening upon in vivo exposure to cocaine would alter this population. So here is the firing of these neurons that would project their axons to the RMTG, and they were identified by antidromic stimulation, is called, basically placing a stimulating electrode here in the RMTG, giving a stim, and identifying whether this backpropagating action potential was indeed present in the recorded neurons. So we could discriminate different neuronal populations like this. What we find is basically the following. Cocaine experience drives an increased activity in vivo of these neurons. So again, not only a passive single injection of cocaine drives the bidirectional activity in RMTG projecting, But even an experience in vivo of these animals drives this hyperexcitability in cocaine-exposed mice. So second hint, saying that this pathway, LHB to RMTG, seems implicated in cocaine effects. Now, one thing I'm not going to go into the details that we found is that this hyperexcitability here seemingly is very important for some aspects of cocaine intake. It does not play a role in cocaine-induced place preference, and it does not play a role in cocaine-induced locomotor activity. So these are classically viewed as reinforcing rewarding properties of cocaine, right? However, if we start looking at other aspects, and especially what we call the depressive-like states after cocaine exposure, what we find is that this connection over here seems to be crucial for depressive symptoms induced during cocaine, that happen and emerge during cocaine withdrawal. So you could raise your question and ask, what are you talking about when you say depressive-like symptoms? I mean, how do you analyze them? So there are a range of several behavioral models that allow us to see the behavior, to test the behavioral despair of the animal, to test how the animals fight against a threatening condition. The forced swim test is one test we used. The tail suspension test is another one, and I'm going to go more into the details afterwards. So this is a work of Frank, a postdoc in the lab, and together with him we put down the following prediction. Negative symptoms during cocaine withdrawal relies on a reorganization of input-specific connectivity in the lateral habenula. So not only it requires this projection, but it requires also some input that are really specific and target a specific neuronal subpopulation in the LHB. And what we did was to focus on this basal ganglia projection that I told you in the beginning. This entopeduncular nucleus, or globus pallidus internal capsule, more in humans, that projects, has been shown to project monosynaptic inputs onto lateral habenula. And remember that it has been shown that EPN inputs, they can co-release glutamate and GABA. Now, but why the EPN? What has anything to do with drug intake or cocaine effects? So one important information is that EPN neurons can encode aversive stimuli, as well as LHB neurons do. That EPN-driven excitation onto the lateral habenula increases in depressive-like symptoms. So there are instances indicating that the drive, the excitatory drive from the EPN is increased when the animal is feeling in this depressive-like state. And then cocaine modifies EPN neuronal activity, and this has been shown both in monkeys and in rodents. So altogether, these two, for us, set the ground to kind of predict that, or to test the hypothesis that during cocaine withdrawal, if depressive-like symptoms emerge, this will be a fundamental circuit engaging these depressive symptoms. So if we were right, we asked the question whether or not the EPN would connect or synapse onto neurons that specifically project to the RMTG and not others. Because if they project to neurons projecting to the VTA, then the circuit specificity for the effects of cocaine will be lost. Yes?
So has it been confirmed that the LHB inputs to RMTG don't have collaterals to the VTA? Because I was under the impression from that rabies virus channel rhodopsin experiment that there might be collaterals.
So in our hands, we have that if we co-inject the retrobits in VTA, same time, VTA and RMTG, two different colors, we do see almost non-overlapping. A critique might be the fact that we are using retrobits. So maybe fiber en passant may also take the retrobits anyway, but still, we have a pretty serious discrepancy between the two neuronal populations. So arguable that there might not be collaterals between the two neuronal populations. However, as Garrett and Alice have shown, there are neurons within the midbrain, within the VTA, that are GABAergic. And therefore, the VTA projection is not the perfect way to test it, because it's structure-specific, but not neuronal-specific. So in a second, maybe I'll convince you that this is true. So what we did, indeed, to test whether this EPN projection would target neurons in the lateral abenula, projecting axon to the RMTG, we employed a Rebi strategies where we used these two lines, the DAT-CRI lines expressing CRI only in dopamine neurons, the VGAT IRS CRI line where the CRI is expressed only the VGAT containing neurons. And we used a series of AAV injected in the midbrain in order to, basically with the Rebi's approach, to find whether or not, or which neurons in the lateral abenula would project either to GABA neurons in the midbrain or to dopamine neurons in the midbrain. And on top of it, we injected an AAV channel rhodopsin in the EPN to test whether those identified Rebi's filled neurons would also respond or not to EPN stimulation. So this is the picture, how it looks like. I think you are familiar with it. But so what you see here, and this is the VGAT-CRI example, is that here in green you see all the terminals from the EPN onto the lateral abenula, very nicely territorially segregated into the lateral portion. And then you see instances of red dots over here, which are these neurons, that in this case are LHB neurons sending axons to GABA neurons. Slightly different is the picture we get in the DAT-CRI. So here, red neurons are LHB neurons projecting axons to dopamine neurons in the midbrain. So you still hear the mash of EPN terminals, but the red dots, they are kind of more laterally located. So suggesting already that these neurons over here, they are not in the field of innervation of the EPN. Now what we did was to go in slices, in acute slices, and patch these neurons in the VGAT-CRI mice and in the DAT-CRI mice. And what we did basically is a regular setting, patch clamp recordings, and LED stimulation of these green terminals in order to evoke synaptic currents at a certain holding potential. So here, for example, is a AMPA-mediated current evoked by light. What we find is basically the following. When we patch neurons in the lateral abenula that are projecting their axons to GABA, therefore in the VGAT-CRI, we see that there is a huge extent, and almost 93%, that responded to light. Differently, in the DAT-CRI mouse, where we can identify lateral abenula neurons projecting to dopamine, 71% of them did not respond to light. This suggests to us that there is a preferential innervation of EPN terminals onto neurons in the lateral abenula that project themselves to GABA neurons of the midbrain, likely RMTG, but with less extent to dopamine neuron. So, knowing that this projection is kind of crucial in the negative states induced by drug exposure, we thought that this three-synaptic circuit will probably be promoting specific behavioral effects during cocaine intake. So the step after would be that we wanted to fulfill was the following. If this is true, would be the function of these synapses, EPN to lateral habenula, modified by cocaine. So what Frank has done was to inject the channelrhodopsin in the EPN first. Here is the lateral projection in the lateral habenula, and then to inject animals either with saline or cocaine for 5 days and record either 2 days or 14 days after the last injection. The prediction here will be if this synapse, EPN to lateral habenula, and especially lateral habenula neuron projecting to RMTG, is implicated in cocaine effects, we would see some sort of a change in their efficacy. What we look at to test this was basically what we called a GABA-A AMPA ratio. We overall looked at the balance between inhibition and excitation. What I've told you earlier is that these EPN projections can co-release, at least from the same terminal, or let's put it like that. There are terminals that can release both glutamate and GABA, okay? And indeed, by putting the cell at a certain potential, we can pharmacologically isolate either an inward AMPA response over here or an outward GABA response. This yields basically this GABA-A AMPA ratio of about 0.4. What we find is the following, that 2 days or 14 days after the exposure to coke, this GABA-A AMPA ratio is significantly smaller in both conditions after cocaine. So, how would you translate this? Come on, one would
an elevation and drive onto the LHB neurons.
An elevation in the drive onto lateral habenula neurons. Would you draw any conclusion on which component will be the one changed after this experiment?
Increase of GABA.
Excuse me?
After this experiment?
I didn't hear.
Okay, increase of GABA receptors?
Increase of GABA receptors. Well, we are saying that there is an increase excitatory drive. So here we find a decrease in GABA-A AMPA ratio. So the GABA-A first, the AMPA after, and so either is this component becoming very big, or this component becoming small. Okay?
Or both?
Well, the ratio changes, so one has to predominate over the other in net change. But my point was, as he was saying, the excitation onto the lateral habenula might have been increased, but this experiment tells us nothing in terms of which of the two is changing. We know, though, that seemingly after this cocaine treatment, there is this modification in this change in balance. Now, to test and check which was this component changing, we started looking at more what are the potential regulator of neurotransmission that could change after cocaine. So we started looking into the AMPA receptors, GABA receptors, vesicular transporter for both GABA and glutamate, any pre or postsynaptic partner or effector that could change this balance. So the way we've done that was basically using an AAV tomato in order to identify all the terminals from EPN to the lateral habenula. And then perform this array tomography, which is sort of an immunofluorescence technique that allows us to do that in very, very thin slices, such that we can probe multiple antibodies on the same slice and really looking at the same terminal for multiple protein expression. So what you see here in this picture is the following. The red channel indicate puncta that are related to the EPN projections. So these are terminals or axons from the EPN into the lateral lamella. The green signal is instead staining for the vesicular GABA transporter. So any of you that would like to say in 3 words the vesicular GABA transporter function? Everyone is on the same page. What, you said no? Okay. So vesicular GABA transporter, where is it located? In the vesicles, right? What's the role? It pumps the neurotransmitter GABA into the vesicles. So it replenishes the vesicle with neurotransmitter, right? Then the vesicle will fuse, and then will release the neurotransmitter that will be prefilled, okay? What did we find here was basically the following. In the cocaine condition, at this EPN terminal, there was a massive decrease in the staining of the vesicular GABA transporter. Yet, the vesicular glutamate transporter was not affected. The GABA-A subunit expression was not affected. The total amount of innervation from EPN to the lateral lamella was not affected. So there was a very specific decrease in the amount of this GABA transporter in the lateral lamella after cocaine. So the picture that may arise will be the following. Given what I just said in terms of function of the VGAT, we thought that the cocaine, what it does, especially at these EPN lateral lamella synapses, is that decreases VGAT that is expressed over here, therefore decreases the content of GABA in each vesicle. What would be this producing? It would be producing a rather decreased amount of GABA, rather presynaptically, that will be available. And if you remember, the GABA-A AMPA ratio was decreased, so we concluded that this was mainly due to a reduction in the GABA component, but not on the glutamate component. So, can we test this? So we wanted to check whether indeed there was or not a reduced vesicular content. So we wanted to check at the single vesicle level whether there was a decrease in neurotransmitter after cocaine. How can we do that without losing this input specificity? So we still wanted to check it at the EPN to lateral lamella synapse. So you could be a reviewer and say, well, show it, and how can we show it? Can we record miniature excitatory or miniature inhibitory postsynaptic current and have the results with it? So what are these minis? These minis, they basically translate for quantal release, therefore content of single vesicle. That's it. In theory, we got the answer. But what's the problematic with the minis? The problematic with the minis is that they will come from any type of input that will be impinging on the recorded cell. So the alternative strategy we used was to stick with the optogenetic-driven activation of the EPN input. But then, to shine the light, get the EPN light evoked current, and then substitute calcium with strontium. This is a trick that physiologists use to basically obtain the following. is called the desynchronization of vesicle release. Basically, when you shine light, you release vesicle all at once by inducing your evoked current. So the release of the vesicle will be timed when you give the light. And this is due to the presynaptic machinery that is based on calcium. Now, substituting these divalent ions, you desynchronize this release, and the vesicle fusing becomes scattered all at once. So you can identify each single vesicle that would fuse by substituting strontium with calcium. This is what happened here. This is the light evoked response, and after that, we desynchronize it with strontium, and then you can see the developing here in a small window of these little events. So what they represent is a fusion of a quanta of neurotransmitter from the EPN. So we maintain the input specificity. And long story short, when we do this in the cocaine-treated mice, what we see is that the amplitude of this quanta is actually smaller. So this confirms the idea that when a vesicle is released, is not filled with the right amount of neurotransmitter. I think you can come up with a question here. Are you convinced of what I've told you? Of course not. Right? Can you come up with a nasty question saying, no.
I just don't get how can you be sure that it's released from a single vesicle, even by doing that?
So,
I mean, could it be more than one at the same time?
no. And the reason is based on it should not be more than one. Okay? So there is the quanta release. You can measure it in terms of amplitude of your events. But the certainty on whether or not you have one or two vesicles fusing at the same time, you're never certain because we are not imaging it. So we are not looking at it. In terms of physiological properties, we are getting closer to desynchronize and have single quanta. Yes?
Would you not also have to show that you don't have down regulation of the receptor of post-synapse?
That's a tough reviewer. So for the moment, I try to convince you of something, but any of the data that I show you could very well apply if there is a downregulation of GABA receptors postsynaptically. So if there was a reduced availability of postsynaptic receptors. I told you earlier that we did not have changes in expression of alpha-1 subunit, but functionally I haven't shown you anything about it. So the decrease in the quanta that I've shown you might be very well due to rather less receptors at the postsynaptic level. And the GABA-AMPA ratio will go well with it. So this is what we come out to answer your question. So we bypass completely the presynaptic aspect at this point, and we focus on the dendrites. And we only activated GABA receptors via GABA uncaging. So we looked at these dendrites in lateral abenular neurons. And then in cocaine condition, we checked whether by light evoke uncaging of GABA, we could activate GABA receptors. Well, you can do that. You can fill up your neuron, see here little dendrites, and then start uncaging GABA at this position, and at this position again, and you get these responses that are purely postsynaptic that are blocked by your GABA-A receptor antagonist. What we see here is that I'm showing you only the somata, but the same is valid for the proximal and the distal. Between cocaine and saline, there is never a change in amplitude that is significant. So this, together with the immunohistochemistry, argues for no postsynaptic changes at the GABA-A level, but rather a pure presynaptic changes given the VGAT modification that we detect. So, in summary, what I've shown you here is that at EPN to LHB synaptic, we have a decrease in GABA transmission, but rather no modification at the glutamate level. And that this presynaptic GABA transmission decrease relies on a decrease in the vesicular GABA transporter. So we claim the existence of a presynaptic defect in vesicle filling and recycling. So, if that was true, can we causally demonstrate this? Can we really make sure that our cellular plasticity it is indeed due to only this VGAT modification? So, we basically wanted to go to control this molecular target that we've identified, the VGAT, and try to make or design a rescue strategy to rescue the synaptic phenotype. What is the strategy we used? We wanted to basically play on the VGAT expression. And we reasoned that if we were to ourselves boost the VGAT expression in the cocaine condition, we could normalize the efficacy of these synapses. And so we come up with this. We injected this adeno-associated virus coding for channel rhodopsin again in the EPN, and we did it together with a VGAT DIO virus. So this virus per se will lead to no overexpression of VGAT until CRE will be expressed in these neurons, right? So after the injection, we came up with the saline and cocaine injection, and therefore we gave time for the plasticity to be put in place. So at this point, 2 days after, we know that the GABA-AMPA ratio will be decreased. Only at that moment, we take back the animals and we put them in the stereotaxic apparatus. And we injected them in the lateral avenula with this retrograde vector, CAV2-CRE. What would happen then? The CAV2-CRE will be retro-transported to all the inputs impinging onto the lateral avenula. But it will drive the recombination and the expression, the overexpression of VGAT, only and solely at EPN to lateral avenula synapses. So this is the picture how it looks like. So here in green is the channel rhodopsin expression at the injection site and in the terminals. And then this is the CAV2-CRE driven M-cherry expression in the EPN and in the terminals. And as you can see here, there is a pretty good degree of overlapping in between the two signals, suggesting indeed that this strategy is efficient in providing overexpression of the VGAT. Now, what happens to our transmission there? This is the experiments that were run with the AV control. So here, the CAV2-CRE leads only to M-cherry overexpression. And after cocaine, you can see still here the decrease in GABA-A-AMPA ratio. But then, when these synapses were challenged with the CAV2-CRE after cocaine, then we find that the GABA-A-AMPA ratio was completely normalized and was actually comparable to even the saline conditions. Now, if you were to look at especially this bar, in these conditions, VGAT will be overexpressed. Would you think that VGAT overexpression will lead to even a further potentiation of the GABA transmission or not?
It depends on whether or not the vesicles are maximally filled to begin with.
Say that again?
It would depend on whether the vesicles are maximally filled to begin with.
Yeah.
If they already are, you would see an increase.
We thought exactly the same thing. So we would have expected or predicted that by VGAT overexpression, we would be then anyway increasing a protein presynaptically, boosting presynaptic GABA release. But as he mentioned, probably this is not happening, and this has been actually reported, because vesicles are actually almost maximally filled with the neurotransmitter. And therefore, even if you put more VGAT in it, you can't pump more neurotransmitter of what is in there. However, when you are in a situation in which the vesicles are submaximally filled, and so there is a certain gap, then the VGAT overexpression becomes efficient in pumping more GABA in it. So overall, we think we have enough data to say, well, probably this is the condition that is occurring upon cocaine exposure. Less GABA within each vesicle. Can we push it forward? Now we have a tool that rescues the synaptic phenotype. What about the behavioral conditions? What type of behavior we can rescue, if we can rescue any with it? So that leads me to the idea of looking at addiction from a different perspective and looking at especially phenomenon like relapse or reinstatement from a little different perspective. So it has been suggested that this repeated consumption may be indeed depending on an equilibrium between the aversive state of a drug and the rewarding state of a drug. The more an individual goes into withdrawal, the more has this feeling of low mood, negative states, and depressive-like symptoms. And they emerge, especially during withdrawal, and it has been suggested, although not clearly proven, that that may be one aspect that contribute to how much the person will go and intake the drug again. So the compulsive behavior will be occurring in parallel with a negative feeling that you, as an addict, you want to relieve. So we wanted to test whether or not, first, the EPN to LHB plasticity will play a role to any of these phenotype, but also to test whether there was any connection between the negative states and conditions such as relapse. How did we test this? So initially, what we did was to condition the animals to a regular apparatus. So condition them to a context with cocaine. Then we put the animals in a sort of a withdrawal period where they learn about the context, and then at one point they didn't get any more the drug. And then we test them in the first swim stress. So basically, in this test has been widely used in the depression field, and it tests for this behavioral despair. The mouse is put in there in swimming, but at one point it gives up in swimming because there is no way out, and the rate of immobility translates for how much the animal won't be fighting anymore, reflecting a depressive-like symptoms. Now, in this test, the first thing we did was the following. If our VGAT plasticity is really playing a role in it, would it be possible to predict that the VGAT decrease will be sufficient per se to drive a depressive-like phenotype? So we wanted to understand whether VGAT in the lateral habenula only by playing with it would drive a depressive-like phenotype, independently of the drug. Because that would be like a proof that this machinery not only changes the physiology of that synapse, but also has a very clear behavioral phenotype. So what we did was the following. I mean, we took animals that were fluxed for the VGAT gene. And so we ablated VGAT in the lateral abenula using the CAV2-CRE retrograde. So in this case, you can see here that in the CAV2-CRE condition, the GABA transmission is pretty much lowered, while the glutamate remained intact. So there is an enormous amount of decreasing GABA-A-AMPA ratio. Yes?
Does that have any effect on the general ability of the mouse to walk around?
Yeah.
You might see an effect on the
Good question. I think I don't have it here, but no, the answer is no. So actually we were afraid about it because this is still a basal ganglia output structure, and the locomotor activity in the open field remains constant. And so that does improve the fact that the animal is not swimming because of locomotion problems. But just to give you the framework here, animals injected with the CAV2, now losing the VGAT, they not only lose this GABA component, but they are already in a depressive-like state. So by ablating the VGAT in that structure and that connection, it's enough to trigger these depressive-like symptoms. So can we now go and test our VGAT overexpression strategy in the context of cocaine? So when you treat the animals with cocaine, then you leave them withdrawal for a while, you see that control mice, they express this higher immobility rate. Okay, this translates for a higher depressive-like phenotype after withdrawal. But then this is completely rescued by the VGAT overexpression. So the VGAT overexpression at EPN to lateral abenular synapse not only normalizes the GABA transmission, but also normalizes the depressive-like phenotype. Yes?
Did you combine your VGAT knockdown with cocaine to see if they included each other?
No, we did not. We did not. Sure, that's a good point. Yeah. Well, sure. On the other hand, what can we expect more? Because what would you do in terms of experimental design would be what?
Do the VGAT overexpression, measure the force swim stress, then give them a cocaine application, and give them either force swim stress or maybe tail suspension as a separate measure. When they've got a learning process with the force swim test. Right,
but so you will use as an outcome only the behavior, basically. Right, sure. There is kind of difficult to check the prediction, right? Because we are in a situation in which the VGAT ablation would already wipe out your GABA transmission. So if you are already at a ceiling effect, I mean, it's not anymore an occlusion type of experiment because you're already maximizing that behavior.
But if you've got an increase in how depressed they were, you might conclude that the cocaine works through the GABA mechanism, but also potentially another one as well.
Yeah, I mean, the best would be to do the opposite, right? That you put cocaine in driving this behavior, and then you could play genetically, but this is a little trickier. So per se, the force swing stress is a stressor, right? So you can well imagine that the mouse went through the treatment with cocaine, then he went through withdrawal, and then it has been shown often that stress can be a very strong inductor of relapse or reinstatement to either intake the drug again or reinstate the preference. So it has been largely shown that a stressor or a challenge with a lower amount of drug can lead the animal to spend more time again in the cocaine-paired compartment. Now, the force swim stress, we used it as sort of analogous of a stress inductor. So what we did was to stress the animal in the forced swim stress, then give them a 15 minutes recovery and to dry up, and then we put them back in the apparatus with no drug. So we wanted to test whether they would spend still more time in the cocaine per compartment. So this is just to illustrate that after the swim stress, in the control group, animals have the tendency to spend more time in the cocaine per compartment. But then in the VGAT overexpressed mice, not only we lose this depressive-like state, but we also lose this preference for the cocaine per compartment. So the two things seems to be going hand in hand. But what we find that it was pretty interesting is that when we plot the rate of immobility, the total immobility in the forced swim stress, and the score for reinstatement, we find that they are positively correlating, suggesting that the amount, let's say, the amount in which the animal is actually feeling this depressive state is connected to the fact that the mouse will go and choose the cocaine per compartment. So the thing seems to be strictly related one to another. So, in other words, what I hope I convinced you is that not only cocaine can provide a modulation of this balance between excitation and inhibition at these EPN LHB synapses, but that this has an impact on a very precise circuit downstream, which is mainly on GABA neurons in the midbrain. And that altogether this seems to code for depressive negative symptoms during drug withdrawal and to establish a connection at the cellular in-circuit level between the depressive-like states and reinstatement or relapse. Yes?
Two questions. First, do you think you would see the same plasticity if the way to administer cocaine would not be IP and passive but self-administration? And third, do you think that your VGAT overexpressing mice would not seek for drug in a Q-induced relapse model where they have to press on together?
To be tested, both of them. So this is the direction where we are moving. So there is a report where they use some self-administration paradigm for cocaine and they see modulation of VGAT. So we know something changes at that level. If this applies to the model you are stating, I would say it would make sense, but has to be tested.
So we were shown by Maria Wolf last week about the incubation withdrawal. So how long out have you checked VGAT expression in withdrawal? Would you predict that it might progressively be downregulated to increase this sort of desire to revert to drug seeking to avoid that adverse response? Or is it maximal after a very short period of withdrawal?
We didn't time it up. We know is there early on, like 5 days after the last injection. We know that is there about 3 weeks after the last injection. Now, would we predict is going to come back at one point or the other? Probably, but to be tested because of homeostatic changing. Maybe the synapse would gain, again, is...
So you don't think it would be a genetic downregulation of transcription of that protein?
Yes, but does it tell you that that has to be long-lasting and that's it? I mean, you can have the downregulation in transcription that is still transient. So there, I'll be curious to see whether this is a machinery that is, okay, the GABA-AMPA ratio stays smaller like this throughout, or rather, this is a transient, then the signal is kind of has a repercussion in downstream circuit, and whatever happens in the abenula, then it's at trigger.
There is another class of drug, like a stimulant, well known to induce very strong withdrawal from the opioid. Do you think your model could be expanded to other class of drugs, or it's more specific to psychostimulant? It means that there are two different pathways according to different class of drugs.
We hope would apply to other class of drugs. We are actually now testing some morphine and check whether we do have similar type of modification. We know we have the idea that the model may apply to any depressive-like type of behavior independently of the insult. So this hyperexcitability of LHB neurons may apply with cocaine with a certain pharmacological trigger, but it also applies with food shocks. It also applies with chronic stress. So if the drug in question, say the opioid, drive some of these depressive symptoms, which they do, I would argue that at least hyperexcitability of these LHB neurons would be a common substrate.
So it would be an even more common substrate than for drugs. It would be all of the...
It will be more of a common substrate than for drugs. Like we have some evidences where an aversive experience, if protracted, can lead to depressive-like symptoms. And through another cellular cascade that is not GABA AMPA ratio, but is GABA B, yet the final outcome is hyperexcitability of these LHB neurons.
So Manuel, if you were to do these experiments in the dopamine transport knock-in animals that have a transporter that is insensitive to cocaine, would it still be here or not?
So, I would say probably we'll lose it. So, what Christian is going after, if I get it straight, is the induction mechanism of this plasticity where they may occur. Can be within the lateral abenula, or it's a circuit-mediated effect. So,
and if it's cocaine, is it the inhibition of the dopamine reuptake or serotonin or norepinephrine?
Yeah.
Right? So it's,
I think my prediction will be we are going to lose this form of plasticity. But another prediction I have is probably this is not occurring because of dopamine actions in place in the lateral abenula.
But there's now a serotonin transporter knock-in that is insensitive to cocaine. So that could be equally interesting.
That could be equally interesting because this. Yes.
Dorsal rata, maybe this is actually serotonin dependent.
And we know that we have both neuromodulation impinging on this structure, but we have evidence that the dopamine in this structure is not necessary for this form of plasticity.
Sort of a related question. Where do you think inputs to the EPN neurons are primarily coming from? I think those are sensitive to plasticity. Good question.
Inputs onto the EPN neurons, you mean?
Right.
Can be many, and we did not test. So we know that there is a cortical afferents that are pretty strong. We know, though, that there is a pretty strong dopaminergic afferents onto these neurons as well. And my guess is that there is some dopamine modulation in the EPN that is gating this. But, so this is a subpopulation of these EPN neurons. They are not all of them sending axons to the lateral abenula. And there may be a little segregation on where they receive inputs from. Marina?
You mentioned a study showing that cocaine exposure affected the GABA transporter in another. Where was that done? What brain region?
I didn't hear.
You mentioned that there had been another study showing that cocaine exposure could affect the GABA.
The VGAT.
Yeah, the VGAT.
There is a, yeah, so,
what brain region?
so the VGAT transporter, that was an immunohistochemistry study that showed it in basal ganglia structures, but without specificity which basal ganglia was. And to me, from the picture, looked rather ventral striatal regions.
And was it message or was it protein?
Was the protein.
Protein.
Was the protein.
All right, thanks.
Was the protein.
Do you expect that other terminals that co-release GABA and glutamate might also undergo this plasticity that aren't from the EPN?
Not sure whether it's going to be the same, but I would expect prediction will be that other input will change as well. So why I'm saying that with certainty is because we know that the glutamatergic transmission changes upon cocaine exposure, on top of what we've seen here at the EPN. However, we see that the EPN excitatory component, that stays pretty unaffected. So there might be at least another input in which the glutamatergic site is changing and probably merging to the same direction in terms of hyperexcitation and hyperexcitability. Which input? Don't know, but I mean, the VTA has been shown to be implicated in drug effects, the lateral hypothalamus as well, and so these are all afferents to the lateral alveolar anyway. Yeah.
So you sort of discussed this within the context of drug withdrawal and sort of a depression-like state that's induced, right? In addition to sort of the depression-related phenotype, another sort of hallmark symptom associated with addiction and withdrawal from many drugs and abuses is sort of enhanced anxiety, right? So what I'm wondering is if there's any evidence whether this circuit or the stimulant in general is either playing a role in that, or do you think the anxiety-provoking circuits are sort of dissociated from the vagal circuitry?
So we had to run a little bit this anxiety test after reviewers' comment. We were a little scared about it because it's not super consistent. So we had to set the good condition. We got it to work, and we get cocaine increasing anxiety after withdrawal, which has been extensively reported. And then we had a couple of animals spare, and we looked at the VGAT overexpression. Animals were still anxious. So argument for that will be probably there are something else, like a parallel pathway that may control different aspects. And I mean, maybe the amygdala would play more of a role for the anxiety part than for the depressive, which the lateral alveolar won't play a role with. So I think this is indicating that, okay, we described cocaine addiction, we described many effects and many aspects, but these aspects, seemingly they need to be taken one by one, and probably they have to be allocated to precise circuits that not necessarily are kind of converging or cross-talking. I mean, the compulsion is another thing. I mean, I'm wondering how much the lateral alveolar function will play a role in compulsion in this case. Okay. Thank you.